Welcome to our condensed version of a video lecture on Punnett grids. If you have never worked with Punnett squares ever before, I highly recommend that you watch the other version of this video. It just goes into a little bit more detail, takes more time to explain things. But if you're feeling pretty good about Punnett squares, then this is the version for you. Our guiding questions are: what patterns of inheritance exist in plants and animals, and what is the molecular basis of inheritance patterns? Lots of objectives here. I'm not going to read these to you, but feel free to pause the video and read if you want to read. Otherwise, on we go. Lots of vocabulary. Again, I'm not necessarily going to read all of these. Pause the video, jot down anything that you're not sure of, but do think about diploid. We have two of each pair of homologous chromosomes, whereas in haploid we only have one. So either the one from the dad or the one from the mom, but not both. Haploid cells are gametes. During fertilization, the gametes meet and we go back to diploid. In humans, diploid is 46 and haploid is 23, but different species have different numbers, so don't get stuck in 23 and 46. And of course, Gregor Mendel is the father of genetics. He did lots of super cool investigations with pea plants. Some scientists argue that his results were a little bit too good, that maybe he fudged some of his data. But if we look at these giant numbers, he had such big sample sizes. These are going to help result in some pretty reliable outcomes. And so maybe this is the argument that we should be making based off of Mendel's work. The bigger the sample size, the better the outcomes. How Mendel carried out his investigations with the pea plants. So flowers have lots of super cool pieces and parts. One of the parts is called an anther. The anther is part of the stamen. The anther produces pollen grains, and those pollen grains are sperm cells. What Mendel did was he collected some of the pollen grains off of a flower, transferred it over to the stigma of a flower. The stigma is kind of sticky. The pollen grains get stuck. The sperm go down, down, down into this reproductive structure called an ovary. And the ovary, there are these ovules, and in the ovules there are eggs. And so those sperm fertilized those eggs, and then baby plants grew. We ended up with some baby plants growing in those seeds. Mendel planted those guys, and then voila, was able to look at all of the traits in the offspring of these different plants. Super cool thing about plants, that we can go ahead and take the pollen grains off of one plant and put it onto a totally different plant, but sometimes we can actually collect the pollen off of the plant and put it onto itself. And so we can have self-fertilization, where the pollen goes onto its own flower, or we can have cross-pollination, where we take the pollen off of one plant and add it to the stigma of a different plant. Lots more vocabulary here. If any of these words you're not quite sure about, pause the video, jot them down. Otherwise, I'm going to jump ahead to this example of a Punnett square or Punnett grid problem. This is a monohybrid cross. We're looking at just one trait at a time. There will be another video later where we do some dihybrid crosses, and we'll look at more than one trait all at once. But for now, just one trait. This trait happens to be about some seeds. We have wrinkled seeds and we have smooth seeds. One of the parent plants is homozygous recessive with wrinkled seeds. The other parent plant is heterozygous with smooth seeds. We know that the heterozygous guys are going to display the dominant trait because the dominant allele will mask the expression of the recessive allele. And so that means that smooth must be dominant. I'm going to use big S, capital S for smooth, and then my recessive trait must be wrinkled. I'm going to give that a lowercase s. This heterozygous pea plant parent plant will be big S, little s. Heterozygous means that those alleles don't match. This homozygous recessive guy is going to have matching recessive alleles. And then we just build this beautiful box and put these alleles together. So potential offspring, we could have a big S, little s, or a big S, little s, or some little s's, or some little s's. I would say that the ratio of genotypes is either 2 to 2, or we could reduce that down to 1 to 1, heterozygous to homozygous recessive. Here are those two heterozygotes, and here are those two homozygous recessive genotypes. I could also say that we have some phenotype ratios. The phenotypes will again be 50% or 2:2 or 1:1. 50%, these guys are going to be smooth seeds, and 50%, these guys will be wrinkled seeds. Feeling good? Feeling good. PKU, or phenylketonuria, is a disorder that is seen in humans. It is autosomal. That means it's not on the X and Y sex chromosomes. It is recessive, and so it is expressed only if there is not a dominant allele present in the genotype. So what is the probability of a child with PKU being born to parents where one is heterozygous and the other one is homozygous dominant? Let's write those genotypes. Heterozygous, 1 for each. I'm going to use P's. And then homozygous dominant, I'm going to use, again, P's. When we cross those two, what we get is big P, big P, big P, big P. Big P, little, and big and little. What this means is that there is a 0% chance of a child with PKU, or phenylketonuria, because it is autosomal recessive. The only way to get this disorder, to be born with this disorder, is to be homozygous recessive. But what if I look at here, I have both parents are carriers for the disorder. Carrier is another way for us to describe heterozygotes. So both parents are heterozygous. Both parents carry the allele for phenylketonuria, but neither one of them actually have it since it's recessive. We do this cross and notice that we have a 1 in 4 chance of a child being homozygous dominant. We have a 2 in 4, or half, probability of a child being a carrier. And then we have a 1 in 4 chance, or a 25% chance, of a child being born with PKU. This does not mean that this family is going to have exactly 4 children and one of them will have PKU. Any given child has a 1 in 4 chance of being born with PKU. The last couple traits that we looked at, the seeds of the pea plants and then PKU, are examples of a complete dominance inheritance pattern. Here we're going to look at codominance, where we literally have more than one allele that is dominant, and so they cannot mask each other's expression. Instead, both of them get expressed equally. Human blood type is an example of a trait that shows this codominance inheritance pattern. How this works, the A and B glycoproteins that are expressed on red blood cells, here we see the glycoprotein on B and the glycoprotein on A, they are codominant, and so people who have the A allele and the B allele get both the A and the B glycoproteins on their red blood cells. Interestingly, type O comes from a recessive allele, so people who are type O have to be homozygous recessive. We show that with some special notation since we have multiple alleles, more than 2. There are 3 different alleles that we can choose from. Of course, each human is only going to get 2 of those, but we have 3 to choose from. To show that type O comes from a recessive allele, we have little i's. A's and B's and AB's have these capital I's because it is dominant. Since we have more than one dominant allele, we show the A, capital I superscript A, and we show the B, capital I superscript B. So you can be homozygous dominant or heterozygous and have type A blood. You can be homozygous dominant or heterozygous and have type B blood. You can be heterozygous codominant and end up with both A and B, and then you have type AB blood. So let's look at this example. I have a man with type AB blood, has children with a woman with type O blood. Let's figure out what are the potential blood types of any potential children born between this couple. So this man with type AB blood, we're going to show his genotype IA, IB, capital I's, because these guys are both dominant alleles. And then the woman has type O blood, that is homozygous recessive, little I. When we mash these gametes together, we end up with a 50% chance of any child being born with type A blood and a 50% chance of a child being born with type B blood. And so there is a 1/2 chance of type A and a 1/2 chance of type B. Similar to codominance is incomplete dominance. How these are different. In codominance, you got both of those alleles being expressed. Here in incomplete dominance, we get a blend of the two alleles being expressed. We're still going to use some capitals and some superscripts, some capitals and some superscripts. Our red 4 o'clock flowers here, these are 4 o'clock flowers, have a genotype of CR. Our white guys have a genotype of CW. When we cross these guys, this is my parent generation. The first generation of flowers that we get, we're going to call that the filial one, or the first filial generation, F for filial or offspring, is going to be a little bit of CR and a little bit of CW, but instead of being red or white, we end up with something in between, and that's pink. If I take two of these guys and I cross them together, now I'm going to get what we call the F2, or the second filial generation. We do this a lot with plants and some animals, but not so much with humans, because we don't want brothers and sisters making babies. That's not really healthy or psychologically good for us. But we do it with plants a lot. So this is our F2, or second filial generation. And so I have my pink and I have the other pink, taking those two baby plants from the first generation, crossing them to make more babies. And we end up with CR. A fourth of the plants are going to be red, and then these guys are going to be pink, and then we end up with the last fourth are white. Let's do this example. Let's say that I've got a pink flowered plant and a red flowered plant. The pink guy is going to be that heterozygote where we have the CRCW. And then the red flowered plant has to be homozygous red. When we do this cross, 2 C's and 2 R's, 2 C's and R and a W. And what this means is that I've got 50% of the baby plants should have red flowers, and then 50% of the baby plants should have pink flowers. Let's switch topics here a little bit. Everything that we've been talking about so far has been autosomal, where we have been looking at all of the chromosomes except for the sex chromosomes. In us humans, we have 23 pairs, 46 total chromosomes. The first 22 pairs, and again, this is a totally human-made ordering of chromosomes. We like to sort them by size, and so chromosome pair number 1 are the biggest ones, and then we get down to chromosome pair number 22. These guys are the smallest of the autosomes, the ones that are just normal old body chromosomes. These ones are special, X's and Y's. An individual who is born with 2 X chromosomes is going to most likely be born with ovaries. A human who has 1 X and 1 Y chromosome is more likely going to be born with testicles. Here's a micrograph picture looking at the X chromosome and the Y chromosome. This Y is a tiny little nugget. Bunches of pieces have fallen off over time. And so a lot of the genes that are on the X chromosome are missing from the Y chromosome. So lots and lots of pieces of DNA are lost. One of the super cool things that is on the Y chromosome that's not on the X chromosome is the SRY gene that codes for a protein called TDF, testes determining factor, testes determining factor. If an embryo has this SRY gene, and that starts making the TDF protein, then we're going to start expressing lots of other genes that lead to the development of testes. If an embryo does not have this gene and does not make that protein, then ovaries are grown instead. What that looks like, if we have a woman with ovaries, she is going to have 2 X chromosomes. The male should be XY, 1 X chromosome, 1 Y chromosome. He's going to make sperm where half of them have X chromosomes and half of them have Y chromosomes. The eggs are going to all have X chromosomes. When these gametes, these eggs and sperm, run into each other, there is the possibility of having 2 X chromosomes. There is the possibility of having an X and a Y chromosome, and so any given child has a 50% chance of being born with testicles, a 50% chance of being born with ovaries. What's super cool about these X and Y chromosomes, again, is that there are a bunch of genes that are on the X chromosome but not on the Y chromosome. So not only do the X and Y chromosomes lead to you're born with testicles or ovaries, but also you might be born with some of these traits. So these ones we actually call sex-linked or even X-linked since they're on just the X chromosome. So here is just a nice little image showing how we can have a father. XY chromosomes is going to give X to daughters, is going to give that Y to sons. Mom, if she's a carrier, that means that she's heterozygous. She has 1 normal and then 1 disorder allele. X chromosome to daughters. X chromosomes to daughters. This daughter is a carrier but heterozygous and so probably doesn't have the disorder, whereas the offspring with the Y chromosomes, because there isn't anything on the Y to mask the expression of that recessive, it's more likely that these affected offspring are going to be sons. Because we're going to add one more piece of vocabulary here, hemizygous. So you've probably heard homozygous and heterozygous before. Now we're going to throw in hemizygous, where we only have 1 allele for the genotype. And this happens in sex-linked traits because the Y is missing that piece of DNA that codes for that trait. So let's look at a couple examples how we do Punnett squares with sex-linked traits. Hemophilia is a blood clotting disorder that is on the X chromosome only. It is not on the Y chromosome. And so when we show these crosses, the genotypes, we're going to go ahead and add the X's and the Y's to show that these traits are on the X chromosome. So if mom is not a carrier, then she's going to be big H on the X's. So X superscript capital H to show that she does not have hemophilia. Dad does have hemophilia, and so he's going to have a lowercase h on his X because he does have that allele for hemophilia. We're going to put no H's on the Y because there is no hemophilia gene on the Y chromosome. Dad's genotype is hemizygous, neither hetero nor homozygous. So when we do this cross, there are some children born with, there's the potential for children born with ovaries, and they could all be carriers. So there's a 50% chance of some female carrier offspring. The other children that could be born, there's a 50% chance that any child born is a male and does not have hemophilia, is totally normal, at least in terms of blood clotting. Let's do another one. Let's say that mom is a carrier and dad does not have hemophilia. Carrier is another way for us to talk about heterozygotes. So mom is 2 X's, and then because she's a carrier, we're going to show big H, little h. And then dad does not have hemophilia. Dad is XY. He does not have hemophilia, so he has that normal allele, capital H. When we mash these eggs and sperm up, we get XX, big H, XX, big H, little h, XY with a big H on the X. Remember, the Y has no H's because that gene is missing from the Y chromosome. Same thing over here, XY, but now this X has a hemophilia gene. What this means is that there is a 1 in 4 chance of a child being born with ovaries and totally normal. There's a 1 in 4 chance of a child being born with ovaries who is a carrier for hemophilia but does not have hemophilia. There is a 1 in 4 chance of a child being born with testicles who does not have hemophilia. There is a 1 in 4 chance of a child being born with testicles who does have hemophilia. All of these different alleles that we've been talking about, the dominants, the recessive, the codominants, are produced by a mutation. Mutations are changes in the DNA sequences of genes, and those result in the formation of SNPs, or single nucleotide polymorphisms. SNPs, SNPs, single nucleotide polymorphisms, where we're literally looking at one nucleotide that has many different forms or shapes. An example is sickle cell anemia. We talked about this in a previous unit, where we have a mutation in the DNA means a new codon in the messenger RNA results in a different amino acid in our protein sequence, and that changes the shape of that protein. And so our hemoglobin in this instance, it gets kind of clumpy, which makes the red blood cells sickled. That's bad. Here's another example just showing this is a cytochrome C oxidase protein. That's this guy, the last protein in the electron transport chain of proteins. And notice that at nucleotide number 1, we have sometimes some C's and sometimes some T's. So again, this is a SNP, a single nucleotide polymorphism. Genotypes are the combinations of alleles that we have for a trait. Phenotypes are the physical traits that we can see that are influenced by those genotypes. Sometimes our phenotypes are exclusively genetic, like our blood type. This is just about what genes do we have. Some of our phenotypes, so some of our traits, come just from the environment, have nothing to do with our genes. Learning how to play the cello is about your environment, your experience. It's not about what gene you were born with. Having a scar from knee surgery, again, this is about your environment, not about a gene. Sometimes we have interactions between genes and environment that lead to a unique phenotype. For example, human height. So how tall we are is part of the genes that we're born with, but also depends a little bit on our nutrition and how muscular we get at a young age and other factors. And so sometimes we have these interactions between genes and environment that lead to different phenotypes. Sometimes we can change the phenotype, so we can turn on and turn off genes at different points. The lac operon is an example of a phenotype that has some plasticity. So bacteria have these genes that make lactase, the enzyme that breaks down lactose, but the bacteria only turn on those genes when lactose is present in the environment, because why make lactase if you don't have any lactose to break down? And so we can turn on or turn off these genes, change the phenotype depending on the environment. Another example is this one. This is particularly known as phenology, and this is all about different genes being turned on due to some seasonal changes. And so when temperatures change, when sunlight levels change, these deciduous plants will activate and deactivate different genes, and so different things are being produced at different points in the seasons. Super cool and a little bit stressful when we start thinking about climate change and how that is going to affect the phenology, these seasonal changes in phenotypes, because that could be pretty scary if our trees don't know when to grow leaves. And my friends, we did it. Again, I'm not going to read these objectives because this is our condensed version of the video. We're going to go to class and do big problem sets. So hopefully you're excited for more Punnett grids. Good work today.